So, hi everyone, I'm Martina, and today in my talk I will talk about some of the ideas I'm currently working on as part of my PhD. So, to give you a little bit of a context, interoception incorporates both lower level of processing along with some higher aspects of metacognitive processing. And today I will focus on these specific dimensions. And these dimensions are really important in understanding both psychiatric and neurological disorders. And together with this, I will provide also some concrete examples from our research. So interoceptive dysfunctions, they are really important. They are conceptualized as transdiagnostic mechanisms, as they are shared across many disorders. For instance, here we have affective disorders. They can manifest, as you see, in different ways. For instance, distorted perception towards heartbeats and also towards inspiratory loads. And neuroimaging studies, they often map those processes onto the limbic system with the anterior insula and anterior cingulate cortex mainly driving maladaptive allostatic shifts. So on the other side, we have also some interoceptive interventions. Here we have two examples. Both they use breathing techniques. So we have a slow breathing that can actually achieve an increase of heart rate variability. And another example is the respiration-gated transauricular vagus nerve stimulation. So both of them, they have been shown to somehow to restore dominance of the parasympathetic system and ameliorate symptoms, basically. So with this in mind, our first project aims at investigating respirosception, which is the ability to sense one's own respiratory-related sensations. And we are currently using the respiratory resistance sensitivity task developed by Micah Zallen group and validated in both behavioral and neuroimaging studies. We will subject a cohort of panic disorder patients due to their experience with breathing-related symptoms and, for instance, like hyperventilation. And that's actually the apparatus and the task. So participants, they breathe into this silicone tubing through this mouthpiece. Here there is a motor that drives a wedge into this tubing, thereby increasing the load. And that's trial samples. So participants, they are asked to take 2 breaths. One of them is constricted, and after the second one, they are asked which one was the harder, and they're asked to provide also a confidence rating. While doing this task, we are measuring MEG activity, and we monitor also peripheral signals. So the output from the task looks like that. So we have adaptive staircase procedure on the background that keeps the accuracy level between 70–80%. We can actually fit a psychometric function, which will give us an objective measure of respiratory sensitivity or accuracy. And looking at the confidence ratings, we can also have some information about the metacognitive aspects I was talking about in the previous slides. We hypothesize that our patients, they will show higher sensitivity and confidence ratings compared to the healthy controls. And at a neural level, we would expect also some alterations in directed connectivity within not only respiration-related networks, but also between some key interoceptive hubs. Moving to the neurological disorders, so we all know that epilepsy is characterized by dysregulation of excitation-inhibition balance, and on the other hand, we know that respiration drives spontaneous shifts of cortical excitability indexed by the 1/f slope. So here we wanted to explore whether also a patient suffering from focal epilepsy shows a different modulation of cortical excitability compared to neurotypical controls, and also whether interictal spike timing depends on both excitability and respiration. To give you some details about the analysis, so of course we measured MEG activity in this patient, and we used the sprint algorithm to relate respiratory phases to 1/f slope estimates, and we built also null distributions by shuffling the phase of the respiration. So that's the result from the single case report. As you can see, that pattern is completely different from the neurotypical controls. So here we can see, circular statistic revealed a significant flattering of the 1/f slope within transition phases, these two, which means an increased excitability within these specific respiratory phases. And regarding the interictal spike timing, so there is an increase again of excitability around the generation of spikes, and this also seems rather specific for transition phases. So again, there is an increase of cortical excitability within these specific transition phases prior to the generation of spikes. To conclude, so we would actually, in our upcoming project together with Gianmarco Duma and Luke Wilson, we would like to have like a full picture of respiratory related alterations of the power spectra, so looking at both aperiodic and periodic activity. We would like also to have some information about the trivariate relationship between brain, respiration, and epilepsy in sleep stages, and also we would like also to apply some connectivity analysis, since we will have different patients, so possibly different epileptogenic zones. So I want to thank you for listening, thank the organizers and also my lab here and everyone working at IBB.
So since we're trying to make up for some time, is there one quick question for Martina? Ah, okay.
Hello, nice talk. I was just wondering, with the respiratory tasks that you had, you said you measured different dimensions of respiratory interoception. I was wondering if you have different predictions with the patients and the controls in terms of like metacognition, sensibility, etc.
So since they tend to have like an attentional bias towards those breathing-related sensations, we would expect higher accuracy in detecting loads and also them being sure about that. So yeah, I would say enhanced metacognitive aspects in this kind of population. And this will also be reflected in directed connectivity. So, I mean, like an increase of top-down signaling.